Okay. Hello, everyone. Welcome to our regularly scheduled GlyConnect webinars. I am Zainab Jain, a GlyConnect executive member. And today I have the honor to chair this session. Before anything, I would like to acknowledge that the University of Alberta is located on the 36 territories, the traditional homeland of the First People of Canada. Before introducing the presenters for today, I'd like to mention that at the end of the webinar, we are going to have an EDI form, which the link is going to be in the chat box. And it helps us to understand how diverse our community and the audience are. And the result for the survey is going to be confidential. So if you feel comfortable, please fill that up for us. And as for the presenters, today we are going to have Dr. Matthew Macauley, who is a professor in the chemistry department of University of Alberta, and Dr. Edward Schmidt, who is a postdoctoral fellow in the Macauley lab at University of Alberta. Right, with that being said, I would give it to Dr. Macauley.
Well, thanks, Zainab. And thanks to the whole GTA for inviting me and Eddie to speak. And thanks for the nice invitation. I remember the first time I had to introduce David, and I was really nervous, but you did a nice job. Yeah, so as Zainab said, this is going to be a bit of a tag team. I think it's a new thing. And so it's for myself, I'm going to speak for, I don't know, 30 or 35 minutes, and then Dr. Eddie Schmidt is going to follow up. And the way I'm going to do this is I'm going to describe some tools. I'll touch a little bit on some applications of those tools. And then Eddie's going to really kickstart that and keep going on the applications and what we've found with them. All right, so let me get into the talk and tell you about Siglecs, which our lab is goes all in on in studying. Why can't I advance? Okay, there we go. So Siglec is an acronym and it stands for sialic acid binding immunoglobulin type lectin. And these are glycan binding receptors in our bodies in humans. There's a really good number of them, about 14 of them. And you can see that they all sort of look the same. They have similar parts, though. They have four different parts that make them up. The carbohydrate binding domain, and this is recognizes sialic acid or sialosides, and it does so through a salt bridge with an essential arginine. And that arginine is going to become important in the talk because we often mutate that arginine to abrogate glycan recognition. The second part is a variable number of C2 set domains. And as you can see, this sticks the VSET domain out from the cell surface by varying degrees. The extremes are Siglec 1 that pops way out of the glycocalyx, and Siglec 3, CD33 here, which is kind of right up against the plasma membrane. Then we have the transmembrane segment that embeds the Siglec on the cell surface. And then finally, a cytoplasmic tail that contains motifs that can control immune cell signaling. And if we look, Siglec sort of 1 through 11 are sort of canonically called inhibitory receptors. And that's because these motifs um, inhibit immune cell activation, whereas 14, 15, and 16 over on the right here are capable of activating immune cells through mechanisms we won't get into. So as I've already alluded to, they can control immune cell activation, these Siglecs, and so not surprisingly, they're expressed throughout the immune system. Here on this slide, we see sort of the major subsets of white blood cells in our bodies, and you can see Siglecs really smattered on all of them. And what do Siglecs do? Well, they bind to their ligands, and like I said, they can control immune cell activation. But there's other hints in the literature that they can do more than that. For example, they can mediate cell adhesion, maybe in certain circumstances. They can endocytose cargo. They can induce activation or modulate cells in trans, so in the other cells that they're binding. And sort of the most canonical method is that they can control immune cell signaling in the cell that they are expressed. So, of these four different roles, there's two really common features of these roles, and that is that these are involve transligand interactions. So, in other words, ligands on another cell or particle, and that they're always multivalent. And this is really the name of the game for us, which is Siglecs bind their ligands fairly weakly on the grand scheme of things. If you wouldn't mind, if you wouldn't mind muting your mics, I'm getting a little bit of feedback from people. So it's very multivalent, and so this is how they work in nature, and therefore we want to exploit that and leverage multivalency. So you can see that in the talk. So this goes a little bit more into detail on what Siglecs are doing in the immune system. I'm not going to go through this all. This is from a review I wrote with Gabriel Rabinovich recently, and it's been submitted. So you can see some major roles for Siglecs on these different types of immune cell subsets, controlling very important immunological processes. And importantly, all involving interactions with their glycan ligands. Here's some more specific roles that our lab is really invested in. I'll just giving you an example of things we study. For example, over on the left here, we study how changes in glycosylation can control B cells. So B cells are the immune cell subset that secrete antibodies and protect us from pathogens. And those antibodies need to get better over time, called antibody affinity maturation. So that's a process we study. We've published one paper on that and have another one that is coming along quite nicely. On the middle here, we see roles for a Siglec called CD33 on microglia, that is immune cells in the brain. We've published quite extensively on that. I won't get into the details, but feel free to look at those publications. And we're also really interested in how Siglecs control immune cell signaling. And so we developed chemical biology approaches to answer questions such as which receptors do they regulate? In this case, PhD grad in our lab, Kelly McCord, investigated or dissected the ability of Siglecs to inhibit this large class of activatory receptors called Fc gamma receptors. All right, but these functions are not going to be the topic of this talk. Notably, these are all driven by ligand attractions and really motivate our understanding of going deeper into what the ligands are. All right, so what are Siglec ligands? Well, we know that each Siglec has a preference in terms of the silicide linkage that they can bind. This has been established over the last, I would say, 20 years, predominantly through glycan microarray. And as you can see, some Siglecs have a strict specificity for the type of linkage. Some have quite a broad specificity. And this has been really valuable information, but our lab hypothesizes that there's something much more specific than just the linkage that dictates whether it's a biological ligand or not. So what could be that more specific definition of what a Siglec ligand is? Here's three ideas that we have, and I'll then describe kind of where we're going with this. So the first thing is that it could be a specific type of glycan structure. For example, a sialic acid on a specific class of glycan, an N-glycan or an O-glycan. That's one possibility. And you can go drill down even deeper than that and look at the exact structure. So for example, something I'm going to come to at the end of the lecture is carbohydrate sulfation as a modulator of Siglec ligands. In addition to a specific type of glycan structure, over the past, I would say 5 years, there's been really an acceleration of professional ligands that have been discovered for Siglecs. And you can see this list here. And we still don't really know clearly why these particular ligands serve as, proteins serve as ligands for these Siglecs. It could be that they have specific types of glycan structures or specific densities, but this is really an emerging area. And then lastly, an idea actually that Ajit Varki had forwarded years ago, which is it could be a specific type of density or cluster of glycan that mediates ligands for Siglecs. And this is an idea that the group in Copenhagen, in particular, Henrik Clausen and Naramatsu, have picked up on. And we participated in a really nice study that they have. So you're hopefully going to see that sometime soon. And they found that on mucins, it is specific clusters that can be ligands. All right. So today I'm going to get into some work that we've done developing new tools. And then I'm going to just start by telling you about some things that we've found with these tools, and that's where Eddie's going to pick up on. All right, so let me start with the tools. So, how does the field generally probe Siglec ligand interactions? Well, a Siglec is embedded in a membrane, and hopefully, most of us on this call understand that a transmembrane protein is biochemically quite difficult to work with. You can't take it out of the membrane and that protein be happy. And so the solution that the field came up with in the 90s was to clone out the extracellular domain of the Siglec, fuse it with the Fc of an antibody to create an Fc chimera. And these continue to be used today. These are sold by R&D systems. And what is generally done is that this is then pre-complexed with a secondary antibody. This then provides at least 4 binding sites to capture some of the affinity. And in general, a lot of very important discoveries have been made with this type of approach. So when I started my lab, we wanted to develop sort of our own set of tools and improve upon this. There are some applications for which, for example, this type of secondary antibody is not, we simply cannot use. And so we re-engineered Siglec FCs, as you can see over here, and incorporated a bunch of new features into them. Some of them I won't get into, but notably a strep tag 2 for purification and assembly of this tetrameric, dimeric, sort of octameric system. So enhanced avidity, and in this manuscript, we showed, basically unveiled these and showed that these to be very successful. So over the last 5 years since we published that first paper, our lab and many other labs have used those tools in a variety of applications. For example, Lab has used that with their liquid glycan array. We've used them with standard glycan microarrays. You'll hear a little bit about that more from Eddie. You can use those on cell-based arrays. Eddie will also tell you about probing Siglec ganglioside interactions. These tools can be used in proximity labeling, photo cross-linking, and then we also work with the Clausen lab to use these tools in mass spec applications. So they've been really widely used, and we've given these tools out to over 50 labs around the world. So just to highlight one application of these tools, and this is, I think, something that Eddie's going to pick up on. This is a manuscript that we have under review. And years ago, we started working with Farid, who has a really extensive glycan microarray from glycans predominantly synthesized in Zichen's lab. So we started teaming up with them to probe ligand interactions on the array. And this is sort of a global summary of everything we looked at. I'm not going to get into all the nuanced details of the ligand interactions, but this is really intended to be an atlas where you can go look at, hey, what does a Siglec bind to on a glycan microarray, and where is it expressed? That's also a really important feature. So just really one really simple point I'll mention here, which I think is kind of cool here, is if I just if I just look at Siglec 5 and 14, so these are actually gene duplications. One is an inhibitory receptor, one is an activatory receptor. And if you look at dozens of reviews that are out there on Siglecs, you will see that it is reported that Siglec 5 is expressed on B cells. And indeed, if you buy an anti-Siglec 5 antibody, you can see Siglec 5 on B cells. However, anti-Siglec 5 recognizes both Siglec 5 and 14. And so in this manuscript, we clarify that point to show that it's actually Siglec 14 that's expressed on B cells, and you can deconvolute that at the mRNA level. So there's a bunch of clarifying points that I think this atlas is going to be very useful for the field. All right, but let me get into the part that really excites me, which is our new tools. And the idea here is Siglecs use multivalency. We've captured that with our first generation set of tools, and the question is, can we do better than that? So here was the goals that we set out for ourselves. We wanted to use a platform that was where we could tune multivalency, ideally up to almost hundreds of copies of the Siglec and make it very modular so that we could tune this very readily. We wanted the Siglec to be covalently attached to our platform so it won't fall apart. And the real underlying reason for that is that we ultimately want to be able to inject this platform in vivo and follow where they go. Sorry, every time someone tries to enter, I can't advance my slide. And then I have to go to a different button. So apologize. There it is again. Okay, so the lab works a lot with liposomal nanoparticles. And so the goal here was to display a Siglec off of a liposome. So how are we going to do that? Well, if you want to find out the details, they're all in this preprint that we put out 3 or 4 weeks ago. This work is now under revision. And so the most of the talk remaining is going to be on this manuscript. All right, so the first thing we thought was how can we link a Siglec to a liposome? And naturally, we thought about click chemistry, bioorthogonal chemistry, and indeed, these were the first approaches that we tried. For example, strain promoted click chemistry, and this simply didn't work for us to the extent that we wanted to. There were several problems, including low reactivity at low concentrations, as well as this very greasy molecule appended in a multivalent fashion from the liposome led to a lot of non-specific interactions with cells. So we disfavored that type of chemistry. We could go to random linking through lysines, but we thought that wouldn't be very reproducible or elegant, and we wouldn't be able to control stoichiometry with the lipid. So through working with the Dirtle Lab had published a nice paper, which I'm going to mention in a bit. They started working with the SpyTag spy catcher system and inspired us to use it and collaborate with them on this. And so the idea here is you have this protein from bacteria, and it naturally has an isopeptide bond in it, so a covalent bond between a lysine and an aspartic acid. And so what had been done was to split this out, this beta sheet out, then optimize this for binding, and this then leads to a very, very quick uh, covalent covalent bond between this peptide and the spy catcher, which is basically the protein lacking this beta sheet. And so the nice thing is you can take this very small amino acid, append it to your protein, and now form a covalent bond with the spy catcher. So here's Zainab, and she was the one that introducing this talk. And if her name rings a bell, you've seen her name somewhere. Yes, she has a sister that graduated from the program as well. Is now a postdoc in David Vocadlo's lab. So what Zainab did was she generated a second generation of Siglec probes. And Here, she appended the spy tag to the C terminus. We tried spy tags at different locations as well, but I'm going to focus on the one that really worked for us the best, which is at the C terminus. Okay, so what we need to do is first introduce the spy catcher onto the liposome. So this was a little project in itself, but unfortunately, we're quite good at bioconjugation of proteins to lipids. So Girma, a postdoc, a then PhD student in the Dirtle lab, had optimized a procedure for expression of spy catcher with a single cysteine. It comes out with glutathione from the bacteria, which can be reduced to form a free thiol, and then a simple reaction with a malamide lipid affords the lipidated product. So you can see here by SDS page, after we've site specifically conjugated the lipid to the spy catcher, we have a nice increase in molecular mass that's apparent by SDS page. So these can be purified as micelles, and then these micelles are then used and formulated with other appropriate lipids, such as the fluorescent lipid, and formulated, extruded to a size of 100 nanometers with a precise number of spy catchers on the surface. And this works quite nicely. So this can then just be simply combined with our second generation Siglec spy tag construct, and these spontaneously form covalent interactions. And this, like I said, this can be tuned by tuning the amount of the spy catcher that you add to this formulation. So here's an STS page gel demonstrating that a covalent bond has been formed here between the Siglec FC and the spy catcher. So here's the covalent adduct here. So here's a little bit of validation of these Siglec liposomes. All this data now is going to be flow cytometry. And if you're not familiar with looking at flow cytometry, what you want to look at is on the x-axis, if the histogram is pushed to the right, that means that the liposomes are binding more to cells. So here's an example using Siglecs 7 or an arginine mutant of Siglecs 7. And these are both formulated in liposomes at different amounts. And so what you can see here as we titrate up the amount, we get more and more binding. The arginine mutant, which is sort of that hollow histogram here, doesn't bind at all, and you can quantify all that information. This top concentration amounts to approximately 200 copies of the Siglec per liposome. And here's a head-to-head comparison between Siglec 7 liposomes and our first generation streptactin tetramers. And you can see, as you can see, the liposomes outperform our first generation platform. And the liposomes also, of course, offer a number of advantages, which I'm going to keep discussing in this talk. All right, we wanted to further optimize this, and something that the field has considered, but really not nailed down, is whether a Siglec FC can be masked by sialic acids on itself. So, of course, we express Siglec FCs from Chinese hamster ovary cells so that they have sort of their native glycosylation. Some people do it from 293 T cells, but same thing. And Siglecs are themselves glycoproteins. Some of them are quite heavily glycosylated. And some labs in the world remove the sialic acid after production using a neuraminidase. We thought we would try a different approach. And so we generated CRISPR knockout of CHO cells in which they lack a key enzyme in sialic acid biosynthesis and express these as sialic acid deficient Siglec FCs. Of course, this is our version 2 with the spy tag on it. So these are now looking at Siglec liposome binding to cells from Siglec 7 FC produced from CMAS knockout versus sufficient cells. And this is a logarithmic scale, so it's a very significant shift. So it looks like CMAS knockout for this Siglec binds better. However, when we go through all the other Siglecs, we find similar phenomenon. In fact, and for some of the Siglecs, like 3 and 9 here, we have close to an order of magnitude increase in binding, so it's quite significant. And in fact, the CHO CMAS knockout cells offer several advantages, such as some of the Siglecs are expressed 2 to 5 times more in these cells. So this has really been our expression platform of choice, expressing Siglecs without salicylic acid on them. So Zainab then undertook this massive effort to express all the Siglecs, all their arginine mutants from CMAF CHO cells, and successfully did so. And so next, I'm going to show you some profiling that we did with all these Siglec liposomes on hand. So here's one where we profiled Siglec ligands on immune cells. And this is looking at human immune cells from peripheral blood. So we simply take the blood, we can subset different immune cells through multi-dimensional flow cytometry, and then stain them with our Siglec liposomes. And here's really a lot of data shown at once, but you can go through all the Siglecs, all the immune cells, and basically tell where there is binding. So this is something that we had done previously with our first generation probes in the 2020 paper, but these Siglec liposomes are much more sensitive, and so this is really a really nice array of data that's I think people can go to and look at where Siglec ligands are on immune cells. So one thing that we notice is that T cells, particularly human T cells, have a lot of Siglec ligands on them compared to other immune cells. And so we kind of got interested in this, and we've been working a little bit with Landon Edgar. They just published a paper on some T cell subsets, and we thought we'd go a little bit deeper into a T cell subset we were curious about, and that's regulatory T cells. And so we got some help from Megan Levings at UBC, who's an expert in this area. She sort of advised on the subsets to look at, and so she helped us deduce or figure out a scheme to gate on conventional T cells and regulatory T cells, again from peripheral blood. Furthermore, we also subset these based on naive versus activated. And I'm just showing the results in naive here. And what on the left, what I'm showing you is the binding to regulatory T cells, and on the right, conventional T cells. And some of these differences are quite profound, up to or over an order of magnitude in binding. So the conclusion here is that regulatory T cells have quite high levels of Siglec ligands on them. And I think that that's a point that the field has not really converged on. And I think this has very interesting functional implications that I'm looking forward to following up on. So another advantage of Siglec liposomes is their the facile way in which they are put together. In other words, the modular way that they're put together. So we played in our manuscript, we played a lot of fun games, I'm going to call it. Zainab may not agree that it was fun. It was fun for me watching the research evolve. So one of the things that we can do is put different Siglecs on the same liposome. And so you might say, Matt, why would you on earth would you want to do that? Well, the motivation I think is quite biologically relevant because a single immune cell can express multiple Siglecs. So putting the Siglecs together on the same platform sort of mimics how they're presented in nature. And so from this, we can then look at how these can work together. I'm not going to show any of that data. We'll show a little bit of data here on how we can multiplex with different colors. And this affords the opportunity of using different Siglecs within the same experiment. So these could be very valuable when, for example, you have a precious biological specimen, or for in vivo studies where you want to have really controlled binding. So this is just a little bit of validation with these 3 different colors. We have Siglec 1, 2, and 7, which bind to sort of non-overlapping sets of glycans. We didn't really want them competing for the same ligands necessarily in this experiment. And I'm just showing a quick heat map of them all pre-complex together versus one at a time with the arginine mutants of the other. And so broadly speaking here, we can see that they're working all together the same as if they were just one at a time. So this sort of proves the point that we can use them and multiplex them together. So this really then motivated a much more complex experiment where we multiplex, in this case, just two of them, but inside of a mouse. So here we took Siglec 2 liposomes with Alexaflor 647 or Siglec 7 liposomes with Alexaflor 488 injected into a mouse. I believe an hour later, we removed tissues. And over on the right, I'm going to show you binding to immune cells. And in this case, we saw the Siglec 7 liposomes binding to the CD4 T cells and the Siglec 2 liposomes binding to the B cells. Now, this in itself might, not be that profound, but this is like actually the exact same binding that we got when we performed the same experiment ex vivo. So we can recapitulate what we can do, get in terms of binding to cells in a tube versus in a mouse. So that was very gratifying, and we're doing it in a multiplexed fashion with two different colors. And so this is really quite reminiscent of work that we've already published with Professor Ratmir Durda. And I'm not going to go into great detail, but they use, hope you hope you know of their work using bacteriophage and chemically derivatizing the bacteriophage. And they undertook a very similar study where they were able to chemically modify um, bacteriophage barcoded with spycatcher, and then put our Siglec FC onto that spycatcher. And they undertook a very similar experiment where they injected these into a mouse, took out the white blood cells, and then profiled where the Siglecs were binding. In particular, in Siglec 7 and the arginine mutant of Siglec 7. And something I just want to highlight here is that Siglec 7 was binding to T cells, but not very much to the B cells. So different platform and almost the same results. Siglec 7 really likes to bind to T cells. So because our liposomes are fluorescent, we can also look at where else are they binding. We were looking to try and find some novel parts of the body that would have Siglec ligands, and so we took out about 5 different tissues, and we saw some interesting staining of the brain, which I'm going to show you. So I hope this shows up well. It looks all right on my screen. But basically, what we see here is Siglec 7 liposomes and injected into the mouse, and you see these little punctate staining. And at first, we stained them with different cells in the brain, such as microglia and neurons, and nothing was really co-staining. And then someone had the idea, hey, maybe this is vasculature. And indeed, if you do a co-stain with CD31 in green, here's the co-stain here and blown up here, all the pink signal is coming and overlapping with vasculature. So it looks like Siglec 7 liposomes are staining the vasculature, or in other words, the blood-brain barrier. And we worked with Stacy Malacher. She provided some of her sticky enzyme that destroys mucins. Siglec 7 is known to like mucins. And doing ex vivo staining of brain slices where Siglec 7 really lights up the vasculature. We can see that we destroy this staining if we pre-treat the cells, or sorry, the tissues with mucinase. So this really reminded us, we actually had these results a couple of years ago, but when this work came out by the Bertozzi and Tony Weiss-Corey paper, it was really, we were really quite excited because our results had matched something similar to them, and they reported that the blood-brain barrier has this very thick coat of mucins. And so it really made a lot of sense in terms of what we were seeing. All right, so that's the Siglec liposomes. Like I said, hopefully the work will be coming out, in the next months sometime. And for those on the call that are interested in accessing our next generation tools, you're welcome to reach out to me. Now I'm just going to finish with a small vignette telling you about how carbohydrate sulfation can enhance Siglec binding. This is something that we've really taken a great interest in, particularly because the results have been just so striking that they've really told us, okay, we really need to go in that direction. All right, let's go back to Siglec ligands. And as I mentioned, we know the linkage that they bind to. Um, and I'm just going to tell a little story about Siglec 7. I've already mentioned that Siglec 7 likes mucins, but I'm going to describe something a little bit more than that. All right, so this project started with first PhD graduate in my lab, Jae-Soo Jung, who wanted to investigate what happens when you overexpress carbohydrate sulfotransferases in a cell. How does this potentially enhance Siglec ligands? So what he did is he used lentivirus to overexpress these 8 different carbohydrate sulfotransferases. These were chosen out of all the genes in humans because they put on sulfation on the galactose or the glucanac of N and O-linked glycans. So unlike ones that work on, for example, chondroitin and heparin, those ones we didn't look at. And so here's a little snapshot of what he did. These are the same type of heat map system using our first generation set of Siglec tools. He would probe these cells. And again, today we're just gonna focus on Siglec 7. What you can see is when CHST1 is overexpressed in the cell, they make a huge amount of Siglec 7 ligands. So what does CHST1 do? CHST1 installs sulfate on the 6th position of galactose. And of note, you can also see that CHST2 and 4 also enhance binding, but not to the same degree. CHST2 and 4 put it on the underlying N-acetylglucosamine. So this CHST1 result was really striking, and we followed this up in a second paper, which we published earlier this year. And what Jae-Soo wanted to do is really go into detail of where that sulfation is being displayed from, what types of glycans. And so he made a glyco-engineered set of library where he knocked out complex N-glycosylation, for example, with MGAT1, complex O-glycosylation with Cosmic. He made cells with a double knockout, and he also looked, if you're interested, go to this paper. He made cells that also lack glycolipids and also omonosylation. And actually, a set of cell lacks all 4, which was kind of crazy to make. But anyways, the result with Siglec 7, because I'm just focusing on that for this talk, was very, very clear. I forgot to mention, so now that he has these glycosylation knockouts, overexpress the CHST1 in them. So as I mentioned, the results with Siglec 7 really crystal clear here. As soon as Cosmic was knocked out, complete abrogation of binding. This is one of these, really beautiful black and white types of findings. So sulfated glycans for Siglec 7 are presented from O-linked, mucin type O-linked glycosylation. And I should really mention that these results are very, very analogous to findings from the Klassen and Niramatsu lab, who used similar types of approaches in HEC293 cells. So these studies really, I would say, complement each other. And a number of groups around the world have picked up on these findings from these papers, and have stimulated them to synthesize sulfated glycans and analyze them by standard glass slides, glycan microarrays. For example, the one on the left here, the one on the left here is from the Boons lab. They made a very nice set of glycans. The one on the right here, I think I have a copy paste error on my references. I apologize, but the one on the right here is from the Wen and Wu lab, and the one on the bottom here is sulfated N glycans, and that's from Lai-Shi Wang and Jim Paulson's lab. We contributed to a couple of these different studies, but all 3 of them actually used our first generation Siglec probes. So I would encourage you to go look up these studies. And there's many really interesting findings in them, in particular for the Siglecs. But for those that are in the field, know that glycan microarrays are very, very good at looking at a swath of glycans, perhaps not quite so good at determining precise affinities. And we were really interested in that for Siglecs 7. So this is where I'm going to end off. I'm going to show you a couple more pieces of data looking at some affinities for Siglecs 7. And so this is partly work that we've already published and some that we're advancing to publication. So in this work that was led by Taylor Gray, but also by Christine Labison, PhD student in the lab, with the assistance from research associate Dr. Maju Zhou. We first started synthesizing some non-sulfated glycans, and then working with the class and lab here, in particular postdoc in the class and lab Duong Bui. They use this very, I'll say, sophisticated native mass spec, concentration independent native mass spec approach. And it's a mouthful to say, and I'm not going to try to explain how it works, but they can deduce dissociation constants without even knowing the concentration of a small molecule. And they've validated this with many targets. So using this approach, we measured these two ligands for Siglecs 7. And so here are the findings here. This value was published in the original ACS Central Science paper, and this second value was published in our work that was just published a couple months ago. And so what these results already show you is that Siglecs 7 really prefers this beta 3 gal linkage, linkage between gal beta 1,3 to GalNAc, as opposed to a type 2 LACNAc structure. So this is very consistent with ours and many other groups' findings that Siglecs 7 likes O-glycans. Then Maju and Christine started stitching on some sulfates. And so here's just a touch of unpublished data that we're writing up now. And so we made a type 2 lacnac with sulfate on it. And interestingly, this didn't really increase the affinity for Siglecs 7. However, putting the sulfate on this gal beta 1,3 GalNAc scaffold now greatly enhanced affinity. So I think it can be, we don't have structural information on how this works, but clearly this different underlying presentation could position that sulfate differently in the, I'll call active site of Siglecs 7. And that's something that we're continuing to pursue with a broader list of O-glycan structures. So I'm going to stop here. I told you about these Siglec liposomes that have all these cool applications that standard approaches do not have. And I told you about how carbohydrate sulfation enhances Siglec ligands on cells. So here is the lab. I think this was last fall. I've mentioned the really key players in my lab, but I do want to thank all of them for their help. I did give shout outs to most of the collaborators. And I also want to really thank funding agencies. In particular, GlycoNet was a really early funder of this work, actually continues to fund this work. And so we're really thankful for them, along with CIHR and NSERC. So I'm going to stop here. I'm going to stop sharing my screen. I'm going to pass on the mic to Eddie. And then I think the plan is that we're going to take questions after Eddie is done.
Thank you very much, Dr. Macauley, for your great presentation. Next, we're going to have Dr. Schmidt.
Okay, hello everyone, and thank you, GlycoNet, for the opportunity to speak, and thanks to Dr. Macauley for setting everything up, so I don't have to do too much intro. And yeah, I'd just like to get into sharing some of the findings we've had with the tools we've developed. So as Dr. Macauley nicely explained a lot about the details of Siglecs, I'd just like to come back to one point about Siglecs is that basically you have a scenario where either two cells meet or a cell and a particle meet, one that has Siglecs and one that has Siglec ligands. They then bind, and then something cool happens. So some of those cool things that can happen are the Siglecs can facilitate holding the immunosynapse together through cell adhesion. They can help to bring in extracellular particles, such as EVs or viruses, and this is very famous with HIV and Siglec-1. They can also help to communicate to cells through modulating cell signaling pathways, and they can do that either on the same cell or on a different cell. But regardless of what function the Siglec is mediating, it all begins with the binding. And that's where most of the work that I'm going to talk about today is about understanding Siglec ligands. So as Dr. Macauley alluded to, Siglecs can be very challenging to study. And one of the reasons for this is if we take a cell like a monocyte, you can see here there are 5 different Siglecs expressed on this monocyte, and it's interacting with the glycocalyx over here. So how do you figure out which Siglec is interacting with which ligand? And that's kind of where my work focuses. And to quote Nobel laureate Carolyn Bottozzi and one of her students, Benjamin Smith, research in the field of glycobiology hinges on accurate methods for identifying and quantifying glycans in a sample. So I'm going to talk about 2 tools that we use to do that, the Siglec FC chimeras, which Dr. Macauley talked a lot about, as well as Siglec ligand liposomes. And these are very similar to the Siglec liposomes, except in this time we put the ligand on the liposome as opposed to the Siglec itself. And I'll start by talking about the Siglec FC. So there are 3 main approaches that we use in our lab. One is a cell-based glycan array. We also have a cell liposome assay, and then the bead liposome assay. And these have been used in quite a few publications, so it's nice to see our tools work not only in our own hands, but also in other groups' hands. And I'll talk a little bit about how we recently used our cell-based glycan array to screen natural Siglec ligands and learn more about how post-glycosylation modifications affect Siglec ligands. So the way this assay works is we have our fluorescently labeled streptactin that we then pre-complex with our Siglec FC to make an octamer. We can then add this to a cell and then incubate them together and then analyze it by flow cytometry. And here on the left, I just have kind of a representative histogram where the pink histogram represents the wild type Siglec. And as Dr. Macauley mentioned, we have arginine mutant controls, so we can really determine how much binding is non-specific or non-sialic acid specific. And this is kind of analogous to an isotype control for more traditional flow cytometry. So using this approach in collaboration with Verid and the Laurie West group, we were able to screen for the ligands of Siglecs on genuine human spleen tissues. And we used the entire human Siglec family here. And to complement this, we're not just interested in where the Siglec ligands are, but we're also interested in where are the Siglecs themselves. So using the same 10 biological replicates, we measured the Siglec expression and noticed a couple interesting things. I think I could talk about this slide for a while, but I'll just try and highlight a few. One is that if we look on the left over here, we can look at the Siglec-1 ligands, and we can see that all of the subsets of immune cells that we looked at have Siglec-1 ligands, particularly classical and non-classical monocytes. But if we look at all these immune cells, there isn't any that really express Siglec-1 under healthy conditions on the subset of immune cells we looked at. We can also look at B cells, which is interesting because CD22 or Siglec-2 is famous for being masked on B cells, meaning that it is engaged with ligands on the B cell, which makes it harder to engage with ligands in trans. And we see that B cells do have a lot of CD22 ligands, and they also express CD22 very highly. So that's congruent with what's previously known. We can also look at Siglec-7, which is a big focus of a lot of studies due to its role in immune evasion. And we can see that a lot of cells have Siglec-7 ligands in it. And it's also expressed by a lot of different types of immune cells. And the last point I'd like to raise is that monocytes, both classical and non-classical, have a lot of Siglecs, and they also have a lot of Siglec ligands. And I think that raises a question of how masking of a Siglec may play a role in regulating that Siglec. And that's something that we look forward to kind of digging into a little bit more, the relationship between the cis ligands and the ability of a Siglec to bind in trans. Another part of the study was looking at how the simple modification of adding a hydroxyl group to the 5' position of the of the sialic acid affects Siglec binding. And to do this, what we did is we engineered some U937 cells, the same cells that Dr. Macauley mentioned had the CHST1 studies on them, and they're a monocytic cell line. And when we overexpress SEMA, we can then convert the glycans from Nu5AC to Nu5GC, and we can use our cell-based array to assess how this affects the binding of Siglecs. So in the right or the top right here is some of the more raw data. And to summarize it a little quicker, we look at here the ratio between the binding between the Nu5AC and the Nu5GC cells. And we can see that Siglec 1, 9, and 15 really prefer Nu5AC glycans instead of Nu5GC, whereas Siglec 10, 8, and 5 really prefer Nu5GC rather than Nu5AC. And a few Siglecs like CD22, Siglec 3, 6, 7, and 11 didn't really have a preference. So that's a little bit about our cell-based glycan array. Now I'll talk about our Siglec liposome platform. So when I first started my PhD, Dr. Macauley and I had a chat and looked at how glycolipid-Siglec interactions were typically studied. And they were typically done in an ELISA, where glycolipids were just non-specifically adsorbed to a surface. We thought that's not a really great representation of the glycocalyx. And I say liposomes are an ideal platform, and I put ideal with an asterisk, because they do recapitulate the lipid bilayer, which makes it more natural. However, we can't say it's a real glycocalyx, and we're definitely missing some nuances in the structure of the genuine glycocalyx, but I think it's a big step up from just a hydrophobic surface. So using this platform in combination with a new panel of Chinese hamster ovary cells, which we engineered to overexpress Siglecs in their membrane. So now we have the Siglec in its natural environment, as well as the glycolipid in its natural environment. We could then mix our cells with our fluorescently labeled liposomes, and we can stain with an antibody to make sure that we're gating on cells that have the Siglec and measure the binding through flow cytometry. And using this approach, we screened the entire human Siglec family against a panel of 9 commercially available glycolipids. And we found some cool things. I think most importantly, we were able to reproduce a lot of the known interactions, such as Siglec 4, Siglec 5, and Siglec 10, and Siglec 1. But the one I want to talk about today is with Siglec 6. And traditionally, Siglec 6 ligands have been kind of mysterious. No one's really been able to lock down what the Siglec ligands are. And we think that we may have found one reason why this may be. And that is that Siglec 6 seems to bind glycolipids really well. But what was really interesting to us is it seemed to do it independent of its canonical arginine residue, which was something unique to Siglec 6. So we followed this up a little bit, kind of passively while we were doing other things. And it really took off when we started collaborating with Elisa Fada's group, who, when we looked at the structure of Siglec 6, we found that Siglec 6 has this tryptophan residue, which is a little odd to have such a hydrophobic residue poking out into the solvent. And what was more interesting is Siglec 6 was the only Siglec to have a tryptophan in this position in the human and the mouse family. So they proposed that this tryptophan might be important in binding to the liposome. So we did some studies where we used the assay I described previously to look at binding to glycolipid liposomes. And we found indeed that when we mutate this tryptophan to an alanine, we lose nearly all binding. We also probed a couple other mutants, and they all had some effect, but the tryptophan was the most striking. So we kind of realized that maybe this tryptophan is driving the binding between the glycolipid and the Siglec, but it's also mediated by the tryptophan binding to the bilayer, which is why in other approaches, such as glycan microarrays, that in our own hands, we've never got Siglec 6 to work. Is why the binding might have been hard to see in the past. And if you want to learn more about this work, it's on the bioRxiv. Please check it out. We were also able to show, using our cell-based or cell-based assay that Siglecs can facilitate the endocytosis of extracellular cargo, such as extracellular vesicles. And this was done with the help of Dr. Kelly McCord. She did the imaging flow here, which we can see that in purple is Siglecs, and in green, this is liposomes. We also did this with EVs. And we had a control here of 4 degrees because when the cell endocytosis is an energetic process, so if we cool the cells down to 4 degrees, they'll still be able to bind, but they won't be able to bring the particles inside. But then you can see when we warm the cells up to 37, we get lots of green on the inside, suggesting that these particles are actually making it inside the cell, and it's being facilitated by Siglecs. And the last thing I'd like to talk about is how we can use these 2 tools together, and that is in our bead liposome assay. So the way this assay works is we take magnetic beads that have either streptavidin or protein G. We can then complex our Siglec FCs to them, and then we can add our liposomes to them, very similar to our cell-based assay. So using this assay in complement with the other data that I just showed, with the help of a fantastic undergrad who I think now just graduated for her Doctor of Pharmacy degree, Su Yan, helped to screen the entire murine Siglec family against the same glycolipids. And we were able to compare human Siglec binding to gangliosides with mouse Siglec binding. And here's kind of a summary of how the human Siglecs compare to their mouse counterparts. But the most exciting one for us was that we found that human Siglec 1 does not engage very well with GM1A, whereas murine Siglec 1, human Siglec 1 does engage with murine, GM1A, but murine Siglec 1 does not. And we used GD1A as a control, and we're able to get some GM1B, which is a more uncommon glycolipid, which has the sialic acid at the terminal position rather than the internal position. And as Siglec 1 is a conserved Siglec, I don't think many of us really appreciate that there can be these subtle differences that might have more profound biological consequences. And so just as a quick summary, I talked about 2 tools to discover Siglec ligands in the Siglec FC and the Siglec ligand liposomes. I talked about some of the assays we can use these with the cell-based glycan array, the bead assay, and the cell assay. There are also a few others that we didn't get to touch, but if you're interested in those, they're described in the publications I mentioned. And here are a few others if you want to take a look at. We're also able to use a combination of these tools to ascribe new biological functions to Siglecs such as Siglecs 6. And hopefully we can apply these tools in a more meaningful way to understand the roles of Siglecs in different diseases such as cancers, allergies, and other chronic diseases. And with that, I'd just like to thank all the wonderful people who made this work possible. And thanks again, GlycoNet, for the opportunity to speak, and all the funding agencies. And if there are any questions for myself or Dr. Macauley, I guess now would be a good time to take them.
Thanks, Eddie, for the great presentation. Before moving to the Q&A session, I'd like to remind one more time about the EDI form that is going to be, the link is going to be in the chat box. Please fill it up if you are comfortable. So about the questions, do we have any questions for Doctors Macauley and Schmidt? You can also type it in the chat box if you are more comfortable. So there's going to be one question.
It's a great talk, Dr. Macaulay, like always. I actually have a quick question. I'm wondering if whether like these tools like that you and your lab is basically develop would have any potentials for further immunotherapy in the future.
Sorry, I missed the first part. What has potential for immunotherapy?
Well, this like the liposome, like different platforms and development, like for immunotherapy purposes.
Yeah, yeah, that's a good question. We've been thinking about applications. I think um, the Bertozzi lab has an interesting paper on bioarchive on these things called ablex, and I know they're pursuing that therapeutically, and it's kind of a similar idea of blocking Siglec ligands. So we've considered, could the Siglec liposomes um, block all the ligands on the cell so that the ligands are not available to do their thing? Could they be used in that type of application? Possibly. I think other approaches might be better for that. Um, where we think they might have profound abilities that are unique to other platforms is when they bind to their ligands on another cell, they may do something to that cell. And actually, part of the work that's in the preprint that I didn't have time to talk about was that Zainab found a role for Siglecs 7 binding to its ligands on T cells. And if you look in the preprint, it activated the T cells. Um, there's probably going to be lots more other applications where a Siglec can do something to another cell. Perhaps it's context dependent. It depends on what other ligands are being co-engaged, and that's sort of the direction that Zainab is going to go next. Um, so in terms of binding and stimulating the cells, I think those have the most applications. But there's other applications we can think of in terms of delivering cargo and that. So yes, I think that they do have applications. So thanks for that question.
Yes, great, thank you.
Thank you. Do we have any other question?
I actually have one question from Dr. Schmidt. Thanks so much for the great presentation. I was actually wondering how much control you have over the presentation of the cyclic ligands on the liposomes, like in terms of like how many like ligands you have. And I actually don't know how you conjugate the ligands to liposomes, but
Yeah, sure. Thank you very much for the question. So in the work I presented today, we use glycolipids, specifically gangliosides, isolated from natural sources such as bovine brain or bovine milk. So the glycans are already conjugated to, they're naturally glycolipids. And then those are purified and we purchase them. So to control the density of the ligand on the liposome, we add different concentrations to our formulation. And in the work, the Nature Communication work from 2023, we have a figure that talks about how we optimized all the different parameters. And actually, glycolipid content was one of the things that was found to be the most important. And for most Siglecs, at around 1 to 3 mole% of the glycolipid, you actually optimize the binding. And above that, you actually start to lose binding. And we think that's because of a density-driven phenomenon. It gets too packed, but we haven't totally locked down exactly what happens when it gets higher than that. Does that answer your question?
yes, absolutely. Thanks so much.
Thank you.
I actually do have another question from Dr. Macaulay. Like you mentioned a lot of like research about like Siglecs 7, which was so exciting. But I was also wondering if you've done also any work on like something like Siglec 11 and its binding affinity to like oligosia and polysialic acid, and how maybe the chain length affects Siglec binding capacity.
Yeah. No, the answer is no, we haven't. I think that that's an open question, a good one. I know that there are people that are really interested in that and pursuing it. So I think at the moment that's not something that we're there's 14 different Siglecs, so you can only you can only do so many things, but I think it is a really interesting question. And probably in the next year or two, you're probably gonna, I'm guessing that we'll get some answers on that.
Thank you so much.
I have a question in the chat for Matthew about multiplex staining. Why in multiplex liposome staining all three Siglecs were the same, but the stained separately? The person's not sure. They can't remember if Siglec 5 also was less when stained separately than the other two.
Yeah, so in there what I kind of went through that data fast, so I apologize, but it's all in the manuscript if you want to take a look. But basically we had controls where they were stained one at a time, or an experiment where they were all mixed together. And basically when they're all mixed together, they kind of behave as if the other ones are not even present. So in other words, they don't interfere with each other. I think that's the main point that we're trying to make there, is that they can be used together.
Thank you. Do we have more questions?
We have one last question if no one else has any questions.
Thanks.
Dr. Macauley, I think you showed that in the cells which don't have the C mass, the binding of the Siglecs are more. But you also mentioned that like you were basically trying to investigate whether cellulation affects the FC binding or not. But I'm just wondering the effect that you're seeing from the increased signal that you're seeing, is that mainly by the cellulation affecting the cyclic ligand binding, or could it also affect the FC spy tag binding to the spy catcher on the liposomes?
I see
And
what
would
you're
you be able to distinguish that basically?
Yeah. I'm not sure we have definitive evidence to rule out that loss of salic acid impedes the ability to form an interaction with the spy catcher, but I don't also think that there's no reason a priori why that would. The spy catcher doesn't really depend on the presence of salic acid to form that interaction. So it's more than likely that it's just the salic acid on the Siglec impedes its ability to form interactions in trans. If if you if you're interested in this area and you go and sift through the literature through groups that have done really extensive work in the area, like Varkey and others, and some of his trainees like Angada, you'll find that they routinely do neuraminidase treatment on the purified Siglecs. Um, and that's presumed to increase binding. But if you look through the literature, we've never found anywhere in the literature that's actually demonstrated that to be the case, where it increases binding. So I think our work is a nice contribution that these cis ligands can impede binding.
Thank you so much.
Thank you. Do we have more questions? Okay, if not, again, thanks all for joining us today, and hope you all have a great week.